Okay, so now that you've finished with your goldfish data and you've collected your respiration rates at various temperatures, we want to talk briefly at just a couple of things to help you start putting together your graphs for your lab report, and also just ways to think about data in general. So I want to start out, we're going to look at your goldfish data or some similar data, and we're going to think about some statistics that can help us when we're looking at various data sets and types of numbers. And then we're going to think about what types of relationships we might see. So how exactly is temperature and respiration rate in fish related? What kinds of relationships might we see? And in particular, we're just going to explore linear or exponential relationships. And then finally, we're going to talk a little bit about your assignment. So the baby graph assignment is due March 26th, so that's the Thursday after spring break. And you can find it on page 83 of your lab manual, and we'll talk about that more in this presentation, okay? All right, so let's start out just looking at some of your goldfish data. So this is actually the data from, I think, the Tuesday lab group, but your data should look very similar. So this is just basically the numbers as you collected them. So we refer to this as your raw data. So you haven't really worked with this in any way. These are just the data you collect. And you don't want to report raw data in your lab report. And we'll talk a little bit about that as we work with this. But when you're looking at just this table, I want you to think about what you can learn from this type of presentation. So just looking at these data, what would your major conclusions be? So think about it for a second, take a look at the numbers. All right, and one thing you're going to notice right away is that because there are 8 different fish and you have a lot of different numbers, it's kind of hard to figure out any exact relationships, right? Because there's a lot of things going on. I would say the main conclusion you could come to from these data is definitely that respiration rate is going up with temperature. So overall, 1630, 65, 4600, 109, 159. Most of these are going up. There might be, I think over here somewhere, there's one that goes down again, the 118. But overall, it looks like respiration rate is increasing with increasing temperature. So that's your major conclusion. Okay. The second thing I think you can figure out from looking at this table is that there's a lot of variability, right? So if you look at 30 degrees, you've got 125 all the way up to 266. So there's a lot of variability going on. So it's hard to really then, just looking at this table, come to more conclusions. We can't really see a lot of the relationships in these data. So we want to think about how you would present that table in your lab report. So you probably want to convert it to a graph, or what we refer to as a figure. So take a minute, you can pause the presentation for a second if you want, and just jot down, make a rough sketch of what you think that graph of those data is going to look like. So make your X and Y axis and maybe sketch out what you think you might see. Okay, so do you have an idea of what you're going to do? So just in general, when you're first looking at your data, so we're going to put our temperature on the X axis, and I'm just going to abbreviate right now. We'll get into what your axes should be labeled in a second, but that's respiration rate. So when we look at respiration rate versus temperature, the first thing you might do is just think, well, let me graph all of the data, right? So you might just put in your table and, graph all your points, okay? And we refer to this as a scatter plot. And this is useful as basically your first step. All right, especially if you have results that you're not quite sure of in looking at the table, what it's going to give you. It's a good way to sort of get the overall view of the data. It's really helpful, let's say you plugged it in and you found there was a point way down here, okay, now I'm going to go back, look at my table. Did I make a mistake? Was that maybe, you put a decimal point in the wrong place, so it might be a typo. So it helps you correct those kind of issues and kind of just generally see what your data is doing. But this is also basically just your raw data. We haven't really worked with this at all. So we want to try to be more concise and summarize it a little bit better. So what you probably want to do instead is, instead of graphing all of the numbers, you probably want to go ahead and take the averages at each temperature. Oops. All right? And so that gives you now a really nice way to look at the relationship. So it's concise, you can see right away what's going on, and depending on what this line does, it gives you some additional information. The question, though, is if we look at that, and then we go and look at our data again, what are we missing? So do the averages tell the whole story? And the answer is you've lost a lot of that information. And in particular, you're going to lose a lot of the information about the variability, about the scatter of your data, and we don't want to lose all of that. So we want to come up with some way to give the reader some more information about the data sets. So here's just a real obvious example. So if you're looking at averages, okay, and let's say you want to take, you have a data set that has 48 and 52, and you take the average and get 50, right? But you could also have a data set that was 98 and 2, and if you take that average, guess what? You're getting 50 as the average. So just saying the average is 50 doesn't tell you anything about these data sets, and these are quite different. Okay, so that's just sort of a real obvious example of how just giving the average is not giving you enough information. Okay, so what we want to try to do is incorporate some measures of variability. Right, and here's a little bit better way of sort of visualizing that the mean or the average doesn't tell you everything. So here are looking at weight and just the proportion of the population at each weight. And you get this nice bell-shaped curve. And the mean for both of these populations is 5 grams, right? But notice that these curves look different. So you've got one that's sort of a narrower, where the results are more clumped together, and one that's a bit more spread out. So you have a little bit more variability in the data. Okay, so we have equal means, but unequal variances, if you want to be statistical about it. So what we've talked about, why are they used? What are some? So here are some frequent ways that you might encounter. So the range is just looking at basically the highest and the lowest at a given number. Right? You're not going to really encounter the range too much probably in biology. There might be a few times where the question you're asking is specifically addressing maybe what the lowest and the highest is, so you might be interested in range. But we're not going to encounter it too much. You will probably more frequently run into standard deviation. So you probably maybe in high school worked with standard deviations. And then the one that we're actually going to use is a measure of standard error, or standard error of the mean. Okay, so how do we calculate these? All right, so this is just going back a step. So this is taking that bell curve that we just saw. And it's just pointing out, so this is sometimes referred to as a normal curve. And if the results fall on this normal curve, it affects the type of statistics you can use. So it's something to just keep in the back of your head. But a lot of characteristics, we're going to sort of assume our data is going to be in a normal curve. Okay? And once you have this normal curve, if you look at the curve itself, notice how there's a point of inflection here. That's going to delineate our standard deviation. So plus and minus 1 standard deviation is encompassing this portion underneath the normal curve. And so if we are plus and minus 1 standard deviation, we're incorporating 68% of our points are within 1 standard deviation. Okay, so that's sort of graphically. And then you could go out to 2 standard deviations. You have another point of inflection, okay? Our equation for standard deviation is shown down here. So this is our standard deviation. And I just want you to note a couple things. Nowadays, our calculators and graphing programs sort of do this for us automatically. But note, one of the things you're putting into this is how far each point away is from the average. So again, we're looking at the variability of our data, okay? So the higher the standard deviation, the more variability you have. Right? This is standard error, or standard error of the mean. And notice we're using our standard deviation, but now we're dividing it by the square root of the number of replicates. So in our case, this is how many fish we're using. Okay, so the numbers, and I think it's 8, 10, something like that for each lab group. Okay, and so if you notice what happens here with the standard error is as your numbers go up, your standard error is going to decrease. Okay, and so what the standard error is measuring is a little bit more than just the variability. We're actually looking at our means, and it's saying how accurate is the average we're calculating. So when you do an experiment, right, we're looking at 10 fish and we're taking the average, but we really want to know something about the population as a whole. So we want to know how close our average is to what the actual average of the population is. And of course, the more fish you measure, if I measured 100 fish or 1,000 fish, I probably have a lot more confidence in my average. So that's what standard error is getting at. So the more numbers you have, your standard error decreases. And that's telling you the range the average is in. So it's the average plus or minus the standard error is saying that if you looked at a bigger population, the actual average is somewhere within that standard error. Okay? All right, so I think this just summarizes. So standard deviation is estimate of the scatter of the overall population. And use it when you're actually asking some question about variability. All right, so a lot of times maybe that's your primary concern. Standard error is a measure of how precisely our sample mean estimates the mean of a larger population. Okay, as sample size increases, standard error decreases. So this is the one we are always using in 105. And I would say for the majority of your career in biology, you probably want to go with standard error most of the time, okay? Regardless of which one you use, you have to always indicate which measure you are using, okay? So since they're different, you have to make sure that your reader or your audience know which measure you're going to use, standard deviation or standard error. Okay, so let me see what the next slide is. Yeah, so let's think a little bit about once we have our graph, right? So we're going to have a graph now, and we're going to indicate with error bars what our standard error is. We want to think about what type of relationship this might show. So what can we learn from making these XY graphs? And so I want to give you two examples. So the first example is, let's say we're looking at an assembly line. So the assembly line can make, I don't know, let's say 5 cars per hour, okay? And no matter what happens, no matter how many cars are already on the lot, that's what it's going to crank out, 5 cars per hour. So if we were to look at the numbers, right, it would be 5, 10, 15, 20, right, et cetera, et cetera. So it's always going up by 5, right? So it's increasing by a constant value each time, a constant number, right? That's not changing. If you were to graph this, if you think about it, 5, 10, 15, 20, what's it going to give you? What kind of line? It's going to give you a perfectly straight line, right? Because it's going up by a constant amount. So this is going to give you a linear relationship. Okay. But our data doesn't always follow a linear relationship. So another way that it might be related is we're going to take the example if you have, let's say, yeast cells, and we're going to look at growth. All right, so a yeast cell is a single cell organism, and when it divides, it splits in two. Okay, so if we start with one yeast cell and it divides, you get 2. They divide, how many are you going to get? 4. And then 4 divide, you're going to wind up with 8, 16. Okay, you can convince yourself and keep going out if you want. So now, how are these increasing each time? Right? It's not a constant, it's not a constant amount. So this is going up 1, 2, now we have 4, now we have 8. So the amount it's increasing is actually also increasing. Okay? So this is actually increasing by a particular percent each time. All right? In this case, we can say it's doubling. Right, and yeast double about every 2 hours, so I would say it doubles every 2 hours. So it's going to increase or double every single time. So if we graph this, all right, now think about it. What kind of line are you going to get? So if you start out with 1, 2, 4, it's starting to go up pretty quickly. So when you graph this, you're going to see more of this J-shaped curve. So this is referred to as an exponential relationship. Okay? All right, so those are our 2 main things that we're going to look at. So we talked about linear. Note the other thing about exponential is that the next number always depends on the previous one. So that's also something you can think about. All right, what do you expect from our goldfish experiment? So have you thought about what you might see? And one thing I want to give you a hint is think about what this Q10 value is telling you. So remember, the Q10 value is the quotient over 10 degrees. So we're going to look at how quickly respiration rate increases over 10-degree intervals. All right, so as an example, if we have 100 opercular beats per minute at 20 degrees, and we have 50 opercular beats per minute at 10 degrees, all right, what's our Q10 going to be? All right, so remember it's the quotient. So if we divide, our Q10 is going to equal 2. Okay, and this is actually, if you look, your textbook has a little blurb on Q10 values. And for metabolic rate functions, you usually will find Q10 values somewhere in the range of 2 to 3. So that's kind of what you expect. So hypothetically, what if you get Q10 values of 2 for, I don't know, let's say 3 Q10 values in a row. So it's doubling every 10 degrees is what this is saying. If a Q10 is 2, it means every 10 degrees your respiration rate doubles. Is that linear or exponential? Okay, so that would be an exponential relationship if that's what you see happening. Now the Q10 values may change, maybe they'll be lower. I don't really know what the exact data is, but I want you to think about how these are all related. So how the graphs are related to what you actually get for the Q10 value. Okay? All right, so if we did find out that our, it seems to be an exponential relationship, the question is how do we deal with data that are exponential? Okay, so here's just again more graphically looking at the 2 different types. So we've got a linear graph, right, and then we've got an exponential. And certainly just looking at these 2, you can see this appears linear, this appears exponential, all right? But another thing we can do is if it actually is exponential, so if this is actually an exponential relationship, there are a few tricks we can do to put it on what's called a semi-log graph. And if we graph it on a semi-log graph, we should then linearize this line. So instead of being J-shaped, on a semi-log graph, it will be a straight line, right? And that would denote that we have an exponential relationship. It also sometimes makes it easier to analyze data because it's a nice straight line and you can maybe predict things a little bit easier. So you'll see this happens a little bit, okay? All right, so how do we actually go about doing this? So what is this? So this is semi-log paper, and it's semi-log paper because if you notice, the y-axis is kind of weird, right? So the spacing here, all right, it starts down here at 1, and it goes to 10, 100, 1,000. So it's just like we were talking about when we were looking at yeast going up. It's not going up by a constant number, but it's going up by exponents of 10, basically. So this is 10 to the 0, 10 to the 1, 10 to the 2, 10 to the 3. All right, we can also think of these as log units, because if you take the log of all these, you get 1, 2, 3, okay? All right, so this is the log scale on the y-axis. The x-axis, though, you'll notice it's just 0, 0.1, 0.2, 0.3, 0.4, okay? So this is a normal scale. So it's semi-log because one of your axes is logged. You may occasionally come across log paper where you would actually log both axes, but we're going to do a semi-log paper. So this is semi-log. We're going to use it when you have an exponential relationship. Okay? Or to see if we have an exponential relationship. So let's just practice a little bit about how the numbering might work on this graph. So let's say I needed to graph 5, 50, and 500, right, on this particular graph paper. So if we look at, again, our range, we're going from 1 to 10, right? And then that's equidistant, so that's the same distance as 10 to 100, is the same as 10, 100 to 1,000, right? So if we're going to graph 5, this is 1, 2, 3, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so there's 10 spots. So 5 is just going to be 1, 2, 3, 4, 5. Okay, did I do that right? And then if we're doing 50, note now we're going to change. So we're going from 10 to 100. So now each division has to be 10, 20, 30, 40, 50. So 50 would be right here. Okay, and then if we're going to graph 500, we're going to go 100, 200, 300, 400, 500. So 1, 2, 3, 4. So that would be right there, correct? All right. So do you understand how you're doing the spacing there? Okay. So one thing to be aware of, so these are the major divisions. The graph paper that you're using is also going to have divisions in between these, okay? So make sure you know how many divisions there are, because where there's only a little bit of space, they may only give you 10 divisions, let's say, but where there's a lot of space, they're going to give you 20. So make sure you know how many divisions there are. So if you have to plot, let's say, 150, right, you need to know how many of those to count to get to 150, okay? All right, so there is our semi-log paper. And if we look, all right, and this is just summing up, and I also put it there to make sure I didn't make any mistakes. So notice there's uneven spacing within each log unit. Here's how you number it, okay? Also note, so in this particular way, what we did was we changed the scaling in order to linearize our data to affect our respiration rate in this case. But you can also do the same thing by, instead of changing the scale, you can also graph by taking the log of each number. So just take, if I was to, you know, I had 50 beats per minute, I would just plug 50 into my calculator, take the log of 50, and you're going to get a number. And you're actually going to plot those numbers, and it'll be something like 0.9, 1.2, 1.4, okay, somewhere in that range. And you're going to graph it on a normal scale, a normal graphing paper. So these are two ways to basically do the same thing. Using semi-log paper, the paper itself takes the log for you because it has different spacing. The good point about using this kind of paper is if I plot, if this is 10, 20, you can easily see what the actual value is, all right? If you're doing it this way and you're taking the log, you sort of lose that information about the original value, but they both should give you the same endpoint, okay? Let's see now. All right, so here's your assignment. You've listened to this video. You're going to go and you're going to do your baby graph exercise, okay? And there's basically two different babies. So there's baby number 1 and baby number 2. And one of them is going to grow with a linear relationship, and the other one is going to show an exponential relationship. So the first thing you want to do is you're going to graph both of them on normal graph paper, right? And you're going to figure out which one grows exponentially, which ones grow linearly, right? And make sure you put all your correct axes labels, right? So that's going to be the first thing. And then you're going to take the baby that has exponential growth, okay? And you're going to graph it using that semi-log graphing paper that we just talked about. So there's some semi-log paper in your lab manual. You may need to go back and just listen again to the end of that presentation if you have trouble putting the numbers in. Okay, so make sure you just double check yourself. So you're going to do a semi-log graph, and then you're actually going to take the logs and graph the logs on normal graph paper. So you can actually see what each of these things is doing to that exponential function, right? So when you do these two graphs, remember that you wind up with a linear line. Okay, if you don't wind up with a straight line, then that means you put the number in incorrectly. So go back and double check. Either double check your logs, or you might have to, sometimes it's helpful to extend the scale a little bit, or check where you're putting in your numbers. Okay? The other thing is just I want to tell you a little bit about how we talk about this. So when you see this as a linear relationship, it's going to get confusing if I have to say linear, because it's really an exponential relationship. It's just showing up as linear. So we refer to this as being log linear. Okay, so it's log linear, it's the same thing as being an exponential. Does that make sense? Okay, so go ahead and try your baby graphs. Remember, it starts on page 83 in your lab manual. And when we come into lab that Thursday after spring break, I'm going to check through all your graphs and make sure you have them done correctly. Okay? The other thing you want to do before Thursday's lab is then watch the second video where I talk a little bit more about how to actually construct your graphs. So we'll talk a little bit about axes and labels and descriptive figure legends, so you're all up to date on that information as well. Okay? So hope you have a good spring break, and I'll see you when you get back.